Synaptogenix. With us for that session is going to be Dan Alkin, MD, President and Chief Scientific Officer. Daniel, how are you?
I'm fine. Good afternoon, Neil.
Thank you. Could you tell us a little bit about your company and get us going?
Sure. Let's begin with the recognition of what one of the major problems today in the 21st century around the world, and that is neurodegeneration. And that's because the epidemiology of the world population is shifting toward the aging population. In the aging population, we already have in the United States alone 6.2 million people who have Alzheimer's disease. In addition, worldwide, we have 50 million people having this disease. By the mid-century, we're going to have 80 million people and 15 million in our country alone. So this is what I would call an insidious epidemic. And it's one of the major frontiers that is confronting the medical establishment in the world today. Yet today, there is no FDA-approved drug that actually treats Alzheimer's disease, the underlying disease, nor is there an FDA-approved diagnostic, despite billions of dollars being invested, and despite hopes that have been raised that a drug might actually treat the underlying disease. There is no such drug. The best we have are drugs that are like Band-Aids. They ameliorate symptoms. They don't actually treat the underlying disease. They may slow down a bit the degeneration results, but they don't slow the degeneration itself. So cognitive decline may slow down 20 or 30%, and that's what we may have heard in the last few years. What is Alzheimer's disease? Alzheimer's disease, by definition of my home institute, which was Neurologic Institute of NIH, where I was a program director for 30 years, is dementia in life, cognitive dysfunction in life, and amyloid plaques and neurofibrillary tangles at autopsy. Those are the pathologic hallmarks. The problem is that when you look at cognitive dysfunction and you try to relate it to those pathologic hallmarks, they're very, very poorly correlated. If we go to the next slide, we can see that not only Alzheimer's disease, but several other syndromes with different causes, different etiologies, also involve the loss of the brain synapses and wiring. The wiring of the brain is degenerating. That includes the nerve cells themselves, which are dying, the synaptic connections themselves, and the long wiring axon processes that link the neurons. So there are a number of neurodegenerative diseases, including Alzheimer's disease, most importantly, but others, such as Fragile X syndrome, Parkinson's disease, stroke, traumatic brain injury. The approach that we take is neuroregenerative. We believe that this is the first time that any company, any major approach has involved regeneration of the wiring. 15 years ago, when we were working at NIH and then at the Rockefeller Neuroscience Institute, where Senator Rockefeller and his family invited me to form a new institute for applying some of what we had learned at NIH. In those years, no one would have expected that you could actually regenerate the wiring that was being destroyed in these different diseases that cause neurodegeneration. But in fact, that's what we found in our studies of many different models of neurodegeneration. We found that we could restore the wiring. In the next slide, we see what are the essential elements of that wiring, the synapses, the connections between brain cells. And it turns out that while amyloid plaques and tangles are not closely correlated with the cognitive dysfunction that the patient has, the dementia that the patients have, the loss of these connections, of which there are trillions in the brain, that loss is closely correlated with the cognitive decline. And that is what has to be addressed. It's the elephant in the room that the industry has not been able to address and is only now beginning, we believe, following our path that we started more than a decade ago. Not many companies, but a couple are trying to follow suit, even with the same mechanistic pathway, to restore wiring. What does that wiring look like? In the next slide, you can actually see wiring in a dish, so to speak. On the left-hand side, you see synaptic wiring, connections between neurons and the neurons themselves that have been degenerated by, for example, soluble amyloid pre-plaque toxic proteins. On the right side, you see the same networks now treated with our lead compound called bryostatin, which is not a statin, but is one of a platform of compounds that orchestrates synaptic growth factors. These synaptic growth factors, such as BDNF, NGF, HGF, IGF, are all orchestrated by PKC epsilon, a particular isozyme that's in the pathway naturally in the brain, linked to a major gene, ApoE4, that's naturally occurring and naturally controlling the genesis of new synapses. This is what it looks like when you treat the degenerating networks with bryostatin or any one of our other platform compounds. You regenerate that wiring. And it doesn't have to be exactly the same wiring. It can be parallel paths that can take over the cognitive function. But this is the fundamental picture of the way we're approaching this disease, and which we believe we will see other companies following this model. And we're starting to see that now. But this is the model we have found is most relevant, and it's directly related to memory storage mechanisms that we identified at the NIH a number of years ago. It's that PKC epsilon synaptic growth pathway that generates new synaptic connections that we can see with 100,000 times magnification using the electron microscope. It's that mechanistic approach that also actually includes preventing amyloid plaques and tau that we believe is responsible for cognitive benefits that we have started to see are clinically meaningful in our early clinical trials. In the next slide, we see a patient who had severe Alzheimer's disease. He was at a mini-mental score of about 2 or 3, and that's indicated by the green circles on the left. When we gave bryostatin, which are those little arrows at the bottom of the panel on the right, with those little arrows indicating doses of bryostatin, we increased that mini-mental score from 3 to about 12. And as long as the target, which is our biomarker, PKC epsilon, was elevated, those are the blue circles, you can see the patient was doing well, not only with mini-mental status exam, but with ADL severe, a clinically useful measurement of how the patient was doing. We, of course, recorded that this patient, while we were observing him over a period of several months, was going from a state of looking at the ceiling and hallucinating to a state where he could choose his food, converse with others, start to swim, engage in a number of activities, and not hallucinating. So he made clinically meaningful improvements with this treatment. So this is a biomarker following the effect of bryostatin on this patient. And we had other compassionate use patients that showed equally dramatic benefits. From this type of approach, we then went to pilot clinical trials, which are illustrated in the next slide. If we can go to the next slide. If you look over on the left-hand side at 0, that's where the patient's baseline values of something called severe impairment battery, which is the metric of choice for advanced patients. And I should emphasize that Synaptogenix, in its pursuit of its neuroregenerative approach, has been looking at advanced Alzheimer's disease. The industry has almost eliminated looking at advanced Alzheimer's disease because they have been so singularly unsuccessful in having any impact on advanced patients. Our patients, on the other hand, have had that impact. And what we see is improvement. So we go from 0 to 3, eventually to 5 and 6. as the treatment continues. And even 30 days after all treatment doses have ceased, which is that dashed line, that fiduciary toward the middle, the patient is still benefiting. So there's a persistent benefit. And this is in the absence of what's called an NMDA channel blocker called Namenda or memantine, because it turns out that our drug actually controls the NMDA receptor and works through it on activity-dependent synaptic growth. For this reason, we don't include patients who have had baseline memantine therapy on board. The controls are the light green and the dark blue, as you see below. And so there isn't any question that there's a major improvement. And I want to contrast that improvement with the slowing of maybe 25 or 30% of the rate of decline that is seen with drugs such as Namenda or the latest Biogen and latest Lilly. These are not improvement over baseline. They're slowing of decline. We believe this is the first clinically meaningful improvement ever seen with drugs that persists for 30 days afterwards. This is the kind of data that has generated a promise for treating long-term patients. So that's why we are now engaged in a 6-month trial, NIH supported, the same approach with now double the number of doses, going after a sustained and even more marked improvement over baseline. In the next slide, we can summarize some of the key findings of our clinical trials to date. First of all, the drug is entirely safe. It's virtually clean. It doesn't, and the dosing protocols that we use, it doesn't have any side effects. We had the benefit of repurposing the drug because originally it was used as an anti-tumorigenic drug in higher doses for 1,600 patients and proved to be well tolerated. We're using considerably lower doses, and it's not just well tolerated, it's totally safe. In addition to this cognitive improvement over baseline, we've seen that this effect persists, and the drug effect increases as the number of drug doses increases. Again, I don't think this has ever been shown with other approaches. This is what's called a trend analysis that demonstrates that the benefit of the patient, this cognitive improvement, is a direct function of the doses that have been given, the dose level. Next slide summarizes a bit further that we don't use Namenda because it's an exclusion criterion, since our drug actually works through the PKC epsilon pathway on synaptic growth factors and also on the NMDA receptors. So blocking the NMDA receptors is not good for the effect of our drug. We have grant support that's currently going over during our trial. It's going over the 6 months. And we are now using patients who are advanced, again, advanced somewhat contrary to what the industry has been forced to do, such as, for example, in the Biogen trial, where they're looking at very early Alzheimer's disease or even mild cognitive impairment. But we're still using advanced patients, but not the most advanced, not the most severe patients. In the next slide, we can get an idea of the pipeline that we are using. The first is we call PUFA derivatives. These are bryostatin-like compounds that we developed ourselves. We have composition of matter protection for those drugs, and they have equipotent and very specific efficacies as bryostatin, and we have, therefore, second and third generation compounds waiting online. These are the indications that we've demonstrated preclinical evidence, extensive preclinical evidence for this regenerative approach, where we actually see the synapses coming back, and then along with the synapses coming back, the cognitive functions being restored to normal. And these are some of the diseases that we hope to go after. The two that we hope to go after the soonest are Fragile X / autism mental retardation and attention deficit disorder with Fragile X, and multiple sclerosis. Again, extensive preclinical models, not only in our own laboratory, but in other laboratories as well. Our most advanced pipeline member is Alzheimer's disease, which is currently in a full phase 2 trial. In the next slide, we can compare what other companies in this competitive landscape might be doing. Although our market capitalization is really quite low compared to some of these other companies, such as Athira or Cassava. Our safety data, we believe, is very extensive. The specificity of how the drug works, the mechanism of action, inducing synaptogenesis and anti-apoptosis, means prevention of neuronal death, we think is uniquely served in our model, indirectly served in others who are using some of the same pathways. And the clinical confirmation that we've seen improvement, not just a slowing of decline, has been demonstrated with conventionally wisdom recognized metrics of cognitive deterioration, namely the severe impairment battery. On this basis, we believe that we have essentially pioneered a field that we're now going into with potentially de-risking the situation by having in the background other compounds as well and other indications that we hope to start on soon. The next slide, just introduce our CEO, distinguished neurologist, Dr. Tuckman, who advises us carefully on designing the clinical trials. Myself, having experience not only at NIH and also as the founding scientific director of the Rockefeller Neuroscience Institute, but also leading teams of multidisciplinary scientists working on this neuroregenerative approach. And the last slide, as a summary, to recap, we're a clinical stage biotech company. We're leveraging bryostatin, not a statin, but a relatively specific, isozyme-specific, and synaptic growth, anti-apoptosis-specific compound, as well as other platform compounds. It should be recognized that these synaptic growth factors are deficient in the Alzheimer brain, such as BDNF. And so is the PKC epsilon, which regulates it. But that deficiency can be restored, as we demonstrated with our preclinical models. And it's that deficiency, we think, that is ordinarily not present in early childhood years when the wiring is being formed. What has been a breakthrough, we believe, that not only we, but we expect others to follow in our footsteps, will lead to a regenerative approach to the loss of brain wiring in a variety of neurodegenerative diseases, Alzheimer's disease being a leading one, but Parkinson's, Fragile X, stroke being other causes as well. We think that this could have a major impact as we go into the 21st century with the insidious epidemic of neurodegeneration.
Astounding. Absolutely astounding, Daniel. Thank you for walking us through that. I just wanted to really quick get one question in before we transition, as we are over on time. So if you don't mind, just one question.
Please.
Do you anticipate starting trials on additional indications this year? And if so, will they be using the synthetic version?
Two good questions. Yes, we do anticipate starting additional indication trials this year. Obviously, we have to be cautious in investing our funds. We are fully funded, although we would love to expand our bandwidth with partnerships and relationships. So that would accelerate our beginning those trials, but that's our intention. And you mentioned a very important factor, that is that we have now exclusive license to a synthetic form of this drug that Stanford University produced and now has exclusively licensed to Synaptogenix, so that we're well positioned ultimately to not only use the natural form which the Cancer Institute has provided, but now the synthetic form, which we believe is totally equivalent and which will give us unlimited access to our drug as we expand our indications.
Absolutely fantastic. Final question. How much are you curling on that bar in the background?
Pardon? I didn't hear that.
How much are you curling on that bar in the back of your workout equipment?
Every day.
That's absolutely fantastic, guys. Inspiring. I love the market for this. I understand the need. I'm sure our viewers do as well. Thank you so much, guys, for being here with us today. Synaptogen.
Thank you, Neil, for your interest and attention.